Thank you for that introduction, and good afternoon, everybody. When we were going through the introductions earlier, I was asked whether it was Reisen or Reisen. I said it's like the old song by, most of you are too young to know it, Creedence Clearwater, Bad Moon Rising. And as a kid, they used to call me Bad Moon, and then it got shortened to Moon. So anybody that remembers that song and has it stuck in their head at 3 in the morning, like I probably will, I offer my apologies now. I'm going to be talking today about new varieties of alfalfa that we're developing at Forage Genetics. And in going over these varieties, I thought it'd be interesting to also talk about some of the breeding approaches that we use, the use of marker-assisted selection and some genetic engineering to develop these traits. And the traits I'll be visiting with you on include fall dormancy and winter survival, improved digestibility with a reduced lignin alfalfa, which will be marketed under the name Harv Extra, and some tannin alfalfas that we're working on, which hopefully will have a similar bloat-tolerant characteristics as we just heard for Sandfoin. Classical breeding is nothing new. It's been done since the Neolithic times when a grower or farmer would keep the best seed of the best variety and plant it again in the subsequent year, and then year after year it develops into the recognizable crops that we have today. So it has a great track record of about 12,000 years. And in alfalfa, it's been especially successful, especially for major traits, and indeed in disease resistance in particular. It's been a great tool for selection and development of new varieties. And since you're only working with the native genes, there are no stewardship requirements, which makes the paperwork and handling and marketing considerably easier. The problem with classical breeding approaches, it takes a long, long time. We have many, many cycles of breeding and selection in the field or in the greenhouse or lab to develop these traits. And we're also limited to the genes on hand within the native population of the alfalfa and some of its nearest relatives. And an example of a close relative where we borrowed some genes would be potato leafhopper resistance in alfalfa, where it was actually crossed to some wild relative, diploid relatives, and 30 years later we actually had varieties that conferred leafhopper resistance. The classic approach typically starts with nurseries like you see here, and at Forage Genetics, currently we put in well in excess of 100,000 plants each and every year in nurseries such as the one here. And we take careful notes on these plants for 3 to 5 years, and then we select the best plants out to use as potential parents for new varieties. The seed we produce off of those plants will go into forage testing for another 3 to 5 years. So the whole cycle from the first nursery to the end product is anywhere from 10 to 15 years. Out of a typical nursery like the one you see here, where we have about 5,000 to 10,000 plants, after 3 to 5 years, we'll be selecting only the best 10 or 20 to go forward with. So it's a very laborious procedure. In breeding for fall dormancy and some of the associated traits with it, we've gone through multiple cycles now, and we have it pretty well defined on how to develop these varieties. Fall dormancy is characterized in a numerical system in alfalfa, and it ranges from a fall dormancy 1, which is very dormant, and up to a fall dormancy 11 type, which would be a type of variety that would be harvested 11 to 12 times a year in areas like Argentina or Saudi Arabia or Mexico. Fall dormancy in and of itself is one of the most important things you can know about your alfalfa when you're getting ready to plant a trial or a field. And the dormancy itself is a response of the plant to day lengths. As the days get shorter, your more dormant alfalfas, which would be your fall dormancy 1, 2s, and 3s, send less of their energy up into growing and more of it down into their roots in order to survive the winter. Whereas your fall dormancy 5s up to 9s and 10s will continue growing, so you have a higher yield potential with those type of products. And this photograph is just one from Davis, California. The photograph was taken in November, and the fall dormancy 9 there is on your right. You can see how much taller it is as compared to the fall dormancy 3 in the middle or the 5. That just equates to more yield potential of that variety in that particular environment. But there are trade-offs with selecting for less dormant alfalfa. And the advantages for more dormant alfalfas include better winter hardiness and overall better persistence, whereas with, but you're giving up that vigor in the spring, faster regrowth, and that later fall yield that you would get with a more non-dormant. So your benefit is you're getting better persistence. The disadvantage is that you're getting lower forage yield. Conversely, with a less dormant alfalfa, you're getting increased plant vigor. The plant's going to mature earlier in the spring, get out of the gates a lot quicker, and you're going to have faster recovery, which may be a good or a bad thing, depending on your location, and increased yield potential. But especially in areas like Saskatoon, you have to be very careful because you do not want to give up lower yield or higher yield for the price of having your stand last another year. And the reason fall dormancy was used for so many years to characterize alfalfa for winter survival can be shown with this slide here. In the green bars here is fall dormancy, and in the blue bars is the winter hardiness of these particular varieties. And these 4 varieties are our standard Czech varieties that we use in alfalfa. And I think you can see why for so many years alfalfa breeders and the industry in general preferred would go with the fall dormancy 2 alfalfa because it had a winter survival of 2 as well. And this was true up into the late 80s when we had some particularly severe winters, and we were able to select plants that had a higher dormancy number, they were less dormant, but still had excellent winter survival. So this chart here shows the same 4 varieties, the older varieties that we use as Czech varieties, Vernal, Dart, 2852, and Archer. And we compare them to some new experimentals that range in dormancy from a fall dormancy 2 to a fall dormancy 5. And if you look at the blue bar there, blue arrow rather, going across, you can see that we now have fall dormancy 5 type varieties that have the same winter hardiness of Vernal at a fall dormancy 2. But if you look at the yield potential, you have all this added advantage here of higher yield, particularly later in the growing season. The genetics can take you so far. There are a lot of other factors involved in winter injury. This is my disclaimer file, or slide here rather. And this just shows a 3-year stand in Wisconsin, and you can notice the plot in the middle there, how badly it was injured over the winter. Temperature is going to be a big driving factor in persistence, as is snow cover. If you have sufficient snow cover, the alfalfa can do fairly well because it has some insulation on top of it. Soil moisture is sort of the Goldilocks of environmental conditions. Too much moisture is bad because the alfalfa will actually pop out of the ground and causing death of the plants. And too little moisture is also an issue. And then there is a bunch of management factors, good fertility, how frequently the alfalfa is being harvested and what age it's at as it's being harvested, leaving sufficient stubble height in the fall to help capture that snow to insulate the alfalfa, and then just a selection of a good variety that has exceptional winter hardiness and disease resistance. Well, this, as I mentioned, most of the breeding that we've done to date on fall dormancy and winter survival has been done through classical breeding programs. And this just shows another nursery here. Each row here has 9 plants in it. Those 9 plants came off of a single parent plant. So we call them half-sieve family. They're brothers and sisters. We know who their maternal side is. We're not as certain about the the father side. And these nurseries are kept in, and they're aggressively managed. They were harvested in where I am in Idaho. We harvest on a 21-day harvest cycle to really put the pressure on the plants. And then we go in and after 3 to 5 years, and we select the plants that are still alive to make into new experimental varieties. Pretty simple. Here's one of those half-sieve family rows I was telling you about. And if you look at the plant that's circled there and that says tolerant, we feel pretty comfortable when we select that plant because its brothers and sisters are still alive. The challenge we have as breeders is sometimes you find a row of plants where everybody's dead or near dead except for that one plant. And the question we have then is that plant truly tolerant, or is it just lucky? Was it in a type of environment in the field where it just didn't have the same pressure as its siblings did? And that's where marker-assisted selection can be a huge advantage and a very useful tool. So with our classical breeding, we're selecting on the phenotype, what the plant looks like. And sometimes in our mind, we see one thing, but in reality, we end up with something else. And unfortunately, in alfalfa breeding, the only way to find out through a classical approach is to combine those plants, wait another 3 to 5 years, and then find out whether you're taking a step forward or you're just holding steady, or in this case, maybe even backing up. So on classical breeding, we're looking at selecting on the way the plant looks, the phenotype of the plant. My old major professor in New Mexico used to say it's a GSP breeding method. This is a joke for the 2 plant breeders in here. It's a grab, stab, and pray. You grab a plant, you stab it into the environment you think is going to express the trait of interest, and then you pray that it works. So you're selecting a lot on that interaction between the genotype and the environment. With molecular markers, we have the ability to identify pieces on the plant's chromosome in the greenhouse or in the lab prior to ever putting it out in the field and selecting for that particular trait. This is a photograph of chromosome 9 in maize and the brown midrib trait. And what they have now is the ability to identify markers that are closely associated to that trait, so they can just chip off a piece of the seed kernel and know before they ever put the plant in the ground whether or not it's going to confer brown midrib expression or not. So it's just a tremendous savings in time and effort for plant breeders. And the marker-assisted selection, as I just said, it speeds up our program. We can do about 3 times the work with markers that we could do with our normal breeding. And actually, the price of the molecular markers have gone down so much over the last 5 years that it's now more expensive to do the actual field testing than it is to do the molecular markers. The disadvantage is that alfalfa is a tetraploid, and that means it has 4 chromosomes instead of 2. So it's a little bit more muddy, and we really don't have as much methodology as they do with corn and some of the other crops. The last 2 traits I'm going to be visiting on are genetically engineered traits. And the big advantage that genetic engineering provides growers, or plant breeders and growers ultimately, is it allows us to introduce traits that would not normally be available in the plant. And a great example of this is Roundup Ready alfalfa, where we have the gene that confers glyphosate resistance, which was recovered out of a soil agrobacterium. Also, tannin alfalfa is bypassed with the higher bypass protein, rumen bypass protein, are also developed this way by introducing genes from other plants. And then the other approach that we use is actually to silence genes that are currently in the plant. And a good example of this is improving forage digestibility through the use of reduced lignin alfalfa. These last 2 traits that I'll be visiting on were both developed in concert with the Consortium for Alfalfa Improvement, which includes the U.S. Dairy Forage Research Center in Wisconsin that did a lot of the sheep and beef studies and dairy studies with this technology. The Noble Foundation out of Ardmore, Oklahoma, which is a large nonprofit organization that actually did the gene silencing in the case of the reduced lignin alfalfa, and then Forage Genetics, which did the breeding. So it's an interesting collaboration. We have a government agency, a nonprofit organization, and a for-profit corporation working together to develop this technology. The reason we're interested in lignin is in a plant, lignin is what keeps it upright. Think of a building and the metal rebar that holds it up. Well, just like that metal's in the building, lignin is in the plant, and it's very tough and it's very non-digestible. As a matter of fact, it's the #1 component of manure solids in alfalfa. And just as if you ever saw a concrete wall that was torn down and you notice that it would have some rebar in it, some of that rebar will actually grab onto the concrete. So if we stretch our analogy here a little bit further, just like concrete grabs onto the rebar, in the plant, that lignin forms cross-links, and it ties up the fiber that normally would be available to the plant. And that fiber is where we're getting our energy, so that's a bad thing. So not only is it a negative for quality in and of itself, it also robs some of that fiber and digestibility that normally would be available to the animal. And this graph here is one that I stole from UC Davis, Dan Putnam, and Steve Orloff. And on the bottom here, we see plant development from the vegetative stage all the way up to post-flower. And the dark line represents forage yield. So over time, as you all know, if you don't cut your alfalfa, it's going to increase in yield as it gets up to full flower. The problem is that more and more of that component of yield is represented by the stem, which is where your lignin is being deposited. So that's why when you look at the dotted line coming down the side there, your forage quality declines as your yield increases. And the challenge for growers is to find the sweet spot where they can maximize their forage digestibility and forage quality without compromising too much on yield or stand persistence. We've been doing a lot of studies over the last 10 years with the reduced lignin alfalfa. I'm just going to cover one from last year that we did. In this particular study, the results are going to be summarized from Idaho and Wisconsin. And this trial was seeded in May of 2015. And the harvest data that I'll be going over was sampled in August of that year. So it would have been the second harvest of the year. And what we did is we went into this field and we harvested on a 3 to 4 day schedule. So we started at day 20 and went up to day 34 and measured the changes in forage quality over time during that growing period. We had 4 varieties in the trial. Harv Extra 118 is a reduced lignin alfalfa. Pioneer 54R02 is a full dormancy 4. And then 2 quality full dormancy 3 types. Legendary HXD and Harv Extra 360 were used as well. When we look at the percent lignin as expressed here in ADL percentage, which is acid detergent lignin, the bottom line there, the yellow line, is your Harv Extra alfalfa. And you can see from day 20 all the way to day 37, there was a statistical difference in the percentage of lignin in the plant as compared to the other 3 varieties, which track very closely together up above. When we look at nutrient-detergent fiber digestibility, the chart flip-flops. So now you see that the highest quality, the most digestible product was the Harv Extra, the yellow line on top, as compared to the other 3 varieties. And we saw about a 5.5% increase in nutrient-detergent fiber digestibility, which translates a 1 unit increase in digestibility would translate into about a 1.7 kilogram increase in dry matter intake and about a 0.25 kilogram increase in milk production. So it's really a pretty dramatic jump. And the other thing we found, and I think this is the real advantage to these reduced lignin varieties, if you see that black arrow, we were observing the same amount of high quality forage at day 34 with the Harv Extra that we saw with the other 3 varieties 10 days earlier at day 23. So we're delaying harvest by 10 days and still getting high quality forage. Well, what happens to your alfalfa over that 10-day period when it's out in the field? It's going to continue to grow and continue to yield. So now you have a little bit more flexibility in your forage. Another way to look at it, again, as yield over time here on the bottom, these are days after harvest, from 20 up to 44, and this is just a generalized graph. Yield, as shown in the blue line, continues to increase, and quality in the dotted line, or in the green line rather, continues to decline. With the reduced lignin alfalfa, we can now delay harvest up to about a week and still get that same quality we were observing earlier, but capitalize on more forage yield. I mentioned we've done a lot of studies over the last several years. And one of those tests was over multiple locations in 2011, where we had 2 harvest systems, a 28-day cycle and a 35-day harvest cycle over the growing season, and we compared the yield that we produced off of these as well as forage quality. And this photograph here is from Touchy, Washington, one of our locations where we had both some Harvexter alfalfa as well as some conventional varieties as checks. And hopefully you can see right down the middle there's a line which represents, I'm not as accurate on this side, which represents the 6 harvests as opposed to the 5 harvest system. And what we saw in the spring, and this is the spring of 2013, was we had much earlier regrowth when you only cut your alfalfa 5 times as opposed to 6 times, which makes sense. And when we looked at the first yield for 2013, we had almost a 1-ton advantage by delaying the harvest with the 5-cut system as opposed to the 6-cut system. So we're getting faster regrowth, better persistence, and higher yield potential with the Harvexter alfalfa without compromising the forage quality. For a grower, to sum up the reduced lignin, one of the most critical decisions for an alfalfa grower or producer is, when do I cut my alfalfa? Typically, it's the same day that it decides to rain in many locations. With the Harvexter alfalfa technology, it gives a grower the luxury of being able to delay his harvest for a day or two or several days without having to worry about compromising his forage quality as badly as he would with a traditional variety. Or he can harvest on his normal schedule and actually have higher forage quality to feed his beef or dairy cows. And the last trait that I'll be talking about are tannin alfalfas. This picture just shows a regular alfalfa in the pale beige color that's been stained with a dye that turns purple in the presence of tannins. And in the middle is one of our condensed tannin alfalfa varieties. And then on the right-hand side is birdfoot trefoil, which naturally expresses high amounts of condensed tannins. Just to show the difference there with these tannin alfalfas. And the reason we're interested in tannins is because of the protein problem in alfalfa. And alfalfa really doesn't have a problem with protein. We have plenty in the field. The problem is that most of that protein gets broken down in the rumen, readily broken down in the rumen, and is excreted in the urine as a nitrogen loss and as a negative to the environment. So what we're trying to do with tannin alfalfas is actually bind up those proteins so that they resist breakdown as readily in the rumen and it can get into the other portions of the cow's stomach where they can be better utilized. The U.S. Dairy Forage Research Center, which is working with us on this project, said that if we could produce an alfalfa that has the same amount of tannins that we see expressed in birdfoot trefoil, we would actually observe about a 60% reduction in the need for protein supplements as part of a cow's ration. And also a 25% reduction in the amount of nitrogen that's excreted into the environment. The USDA model also went on to say that this would translate economically into about a 12% net return for dairies, and alfalfa that gets ensiled has similar microbial activity working on it as it does in a cow's rumen. So we'd be a protection for ensiled alfalfa of about $23 a ton. And then finally, as we just heard, sandfoin, birdfoot trefoil, sisal milkvest, there are a lot of other forages out there that have tannins to protect them, protect the protein, and they are all, as a rule, non-bloating. USDA has estimated worldwide losses of bloat to be in excess of $200 million a year. So there would be a big advantage there as well. These last two traits were part of the Consortium for Alfalfa Improvement, and we've made some really remarkable changes in alfalfa over the years through this collaboration. Over the years as well, more and more people have joined us, and more organizations also are collaborating with us now to develop new and better varieties. That's it.
Excellent. Thank you, Peter. That was excellent. Does anybody have any questions? No questions. Well, I think you did an excellent presentation, and you're obviously very busy there.
Yeah.
Thank you so much,
You're
Peter.
welcome.
Thank you.